Welcome to the second video in the Innate Immunity unit. In this video, we're going to walk through the different cells of the innate immune system and what the general function of each one of those cells are. So this is going to be important as we move into the next video on inflammation, as we talk about how all of the cell functions are going to come together in one large process. Let's go ahead and take a look. All the cells of the immune system develop from what are called multipotent stem cells in the bone marrow. These multipotent stem cells have the ability to become multiple different types of cells that have different functions. And so the step is going to begin by going down one of two differentiation pathways. These multipotent stem cells can either go down the myeloid line or down the lymphoid line. The lymphoid line is going to lead to the development of what are called lymphocytes. Those lymphocytes can then develop further into T cells and B cells. These are the cells of the adaptive immune system that we're really going to be talking about in the next unit, so we're not going to see them much in this lecture. However, I did want to point out that there are these large natural killer cells that are lymphocytes down this line, but are part of the innate system. And this is the only cell of the lymphoid line that is going to be talked about today because it is part of innate immunity and that it functions non-specifically to do its job. For the myeloid pathway can lead to a number of different cell types. So along the myeloid pathway, we get some of our blood cells, so things like erythrocytes, your red blood cells, and also the megakaryocytes, which are going to develop the thrombocytes that lead to things like platelets for clotting. Those are not part of the immune system, and so we're not going to go over the functions of those cells. For immunology, what we care about down the myeloid line are the mast cells, which we'll talk about, and then these myeloblasts. Myeloblasts are still not fully differentiated. Your myeloblasts can become one of four cell types. The first three are what we call the granulocytes. These are the basophils, the neutrophils, and the eosinophils. We'll talk about how those function. And then the fourth are called agranulocytes, and these are monocyte. The monocytes function in the bloodstream, but if they move out into the tissue, they will differentiate further and become what we call macrophages or dendritic cells. And so we're going to walk through all of the functions of these myeloid cells that are part of the innate immune system. It should be noted that all those cells that I just talked about are all called leukocytes, and that's the scientific term for what you often hear called white blood cells, or the cells of the immune system. These leukocytes make up about 1% of the total blood volume, so most of the innate immune cells are circulating in the bloodstream. And as we look at them, you'll see they're in kind of different groupings. So you've got your neutrophils, your basophils, and your eosinophils, which are called granulocytes or PMNs. You've got your mast cells, and then we have what are called the agranulocytes. These are monocytes, remember can develop into macrophages or dendritic cells, and your lymphocytes, your T cells, your B cells that we're going to talk about in the adaptive unit. One thing to note is that leukocyte might sound a little bit familiar to you if you think of the term leukemia. Leukemia are blood cancers, and they're cancers of any of the cell types that we just mentioned. So you can have leukemias that are made up of mast cells. You can have leukemias that are made up of myeloblasts or neutrophils. And those are just a term for all of the blood cancers that could happen if your white blood cells themselves become cancerous. The first cells we'll look at are granulocytes. So granulocytes are called granulocytes because they're filled with little vesicles, so little membrane-bound vesicles, that have various chemicals in them. And those chemicals are going to be released by the cell when it becomes activated. It's those chemicals that do the job of the cell in the immune system. And so you can see the granules in the cartoon versions here. You can also see them in the real cells here under the microscope. They're a little harder to see in your neutrophils. But notice the cells clearly look different. So for your granulocytes, we have neutrophils, which have these big multi-lobed nuclei. So this is actually the nucleus, and have the granules that are much fainter in color. We have eosinophils that have this kind of bilobed nucleus, and that have these red-filled granules. And then we have basophils that have kind of this one big blob of a nucleus that have these purplish colored granules. The reason I point that out is because you can look at these in a blood smear and actually identify them and count them, and that can tell you something about what type of infection you have. Of the three cell types, it's your neutrophils that are the most important. So this is the most abundant leukocyte in the bloodstream, makes up about 50 to 70% of all the circulating white blood cells. And it does two things. It's able to do phagocytosis of pathogens, so it's able to actually pull them into the cell and destroy them. And it will also do killing through cytotoxic chemicals that are found in its granules. So its granules itself contain chemicals that are able to poke holes in cell membranes. When they're released from the cell, those chemicals can kill the bacteria. Eosinophils are involved with things that are killing of helminths, mainly things that are too large to be phagocytized. And basophils are filled with histamine that are supposed to be signaling for those helminth infections. So they release the histamine in response to large molecules, and that recruits the eosinophils over to kill those large molecules. Unfortunately for us, the basophils and the eosinophils, especially in the developed world where we don't have a whole lot of helminth infections, are mainly these days responding to large things that enter the body that aren't actually pathogens. Things like pollen grain or pet dander that people are allergic to. Okay, so if you have kind of seasonal allergies, you can thank your basophils. They're the ones that are signaling that something's wrong, even though realistically nothing's wrong. And this is why you take antihistamines if you have allergies, because those basophils, what's in all of those granules are histamine that's actually causing your allergy symptoms. The next cell are the mast cells. These also contain granules, so those little vesicles that are filled with proteins they're going to release, but they aren't actually considered granulocytes, mainly because they're not circulating in the bloodstream the way the neutrophils, eosinophils, and basophils are. Your mast cells actually reside out in the tissue, mainly in the connective tissue and the mucosa, where they patrol for pathogens as they might come into the tissues themselves. They've got a key role in inflammation, so we'll talk about these cells again, but we talk about the section on inflammation. The granules for mast cells contain both histamine and heparin, which is involved in the inflammatory response. The last cells to be derived from the myeloblast were monocytes. The key trait of monocytes are that they're effective phagocytic cells. So they're really good at phagocytosis and kind of pulling those foreign particles into the cell to digest them in the lysosome. Your monocytes, remember, are going to reside in the bloodstream, whereas if they move into tissues, they're then going to differentiate into either macrophages or dendritic cells. Both macrophages and dendritic cells are phagocytic cells, so they're both involved in the phagocytic process, but macrophages tend to be more important for inflammation, while dendritic cells tend to be more important as what we call an antigen-presenting cell, which connects the innate system to the adaptive system. And macrophages can also present antigen to the adaptive system, they just don't do it as often. And so while they're both very similar cell types, they kind of have their own niche that they fall into in the environment. We'll look at macrophages more in detail in the inflammation section, and we'll talk about dendritic cells more when we get to the adaptive immune system. So all the cells we've been talking about have to have a way that they're able to recognize that something's a pathogen. So how are they telling self cells from non-self, or those foreign cells? The main way they do this is by looking for specific molecular molecules that are unique to pathogens or that are unique to microbes. And they have receptors that are able to bind those molecules to recognize them. So these are things like lipoproteins or lipopolysaccharides that are found on gram-negative cells. There are things like the flagellin protein that made up bacterial flagella, because remember the bacterial flagella was different than eukaryotic flagella, or things like peptidoglycan that makes up the bacterial cell wall. Again, molecules that are unique to the microbes that are not found in humans. This lets us say that's foreign and not self. We call these types of molecules PAMPs for pathogen-associated molecular patterns, or sometimes more generally MAMPs for microbial-associated molecular patterns. The way that our white blood cells recognize these PAMPs is by something called a pattern recognition receptor. So all of these receptors seen here on the cell, these are all what we call PRRs. So they have a specific receptor that's going to bind to peptidoglycan, another PRR for flagellin, another PRR for lipoproteins, so on and so forth. A common type of PRR are called toll-like receptors. And so there's a whole classification of proteins that are toll-like receptors that all recognize a different one of these PAMPs on the pathogen itself. This binding here between the PAMP and the PRR then signals into the cell to affect gene expression. And so this is going to cause changes in gene expression where we're going to start transcribing and translating different genes that are involved in producing those cytokines. Okay, so the voices of the immune cell, so now they're saying, hey, we found this microbe, we found this foreign molecule, we need to do something to it. And this is also then how they induce phagocytosis to start destroying these cells. Phagocytosis is a common method used in the innate immune cells to start removing and destroying the foreign invader microbes from the body. The cells that can do phagocytosis, remember, are macrophages, your dendritic cells, which both come from monocytes, along with your neutrophils. So all those cells can do this cellular eating process. At first, we need to have that connection. So we've got the receptors, remember the PRRs, that bind to the PAMPs on the bacterium. When that happens, it signals for phagocytosis, and so we start extending these pseudopods from the cell, and they're going to reach around until they connect, and they pull that bacteria into a phagosome. The key is the phagosome is then going to fuse together with the lysosome, forming this structure here that's called a phagolysosome. Remember, the lysosomes are the stomachs of the cell. They're where all the digestive molecules are. So when the phagosome with the bacterium or the virus, whatever's been taken into the cell, fuses with the lysosome, all the things in the lysosome now are going to start to digest and degrade and kill that pathogen. So these are things like digestive enzymes such as lysozyme. They're also reactive oxygen species like superoxides or hydrogen peroxides, which can damage the pathogen. And if the pathogen is really hard to kill, sometimes then the macrophage will get activated. So the other immune cells will say, hey, you need some help, and they'll help activate the cell. When that happens, it starts producing reactive nitrogen species like nitric oxide, and that's really deadly and really dangerous and can help kill the pathogens that are in those phagolysosomes. Once the phagolysosome has been successful, we've digested that pathogen into little itty bits, and then those can be released from the cell through exocytosis. This phagocytic process can be improved, which is called opsonization. And so remember, we already mentioned that complement is able to opsonize. That's because if C3b is bound to cells, the macrophages or the other phagocytic cells have a receptor that will bind C3b, and that will trigger this process. Same thing happens with antibody. So molecules that have antibodies bound to them can then be more easily phagocytosed, and we'll look at that more when we talk about the adaptive immune system. The last cell of the innate system that I wanted to mention were the natural killer cells. Remember, these are the ones that are different than everything else because these are lymphocytes. They're from the lymphocytic line as opposed to the myeloid line of everything we've been talking about so far. But they're not part of the adaptive system. Their main job is to identify and kill cancerous cells. So remember, foreign molecules can come from outside the body like pathogens, but they can also become your cells. So if your cells mutate and start doing something that's not self, they become foreign molecules, and it's the job of the immune system to recognize and destroy them. So normal healthy cells on their surface have this molecule called MHC1. This is going to be an important molecule. We'll talk about it a lot when we talk about the adaptive immune system. All you need to understand right now, though, is it's found on all of your normal body cells. And so healthy cells have this molecule. If they do, the natural killer cell here can bind to it and say, nope, yourself, you can go on living, there's nothing wrong with you. But sometimes cancerous cells or cells that are infected with a virus will lose the MHC1. So notice there's no longer MHC1 on the surface of this. That's the signal to the natural killer cell that says you are no longer self, and so they have other interactions that will then occur, and the natural killer cell releases molecules, mainly they're granules containing something called perforin, which can perforate the cell, it pokes holes in the cell membrane, and various cytotoxic enzymes, which lead to lysis of your own cells. And so natural killer cells' job is to kill our own cells when they're no longer functioning like self cells. The reason they're part of the innate system is because they are nonspecific. This isn't, say, killing a heart cell or a liver cell. It's just killing any cell that has lost that MHC1. It's important for you to understand the functions of all the different cell types we've talked about, but you don't need to memorize the amounts of them that are typically found in the cell. However, I did want to point out that it is important to understand why we care what the normal levels are for these different cell types. And it's because when you look to see how those cell types change, it can help to tell you what type of infection you might have. This is the information that you gain when someone orders a CBC, or a cell blood count, or a differential, which will actually give you the different types. And so we can see here, for example, if your lymphocytes seem to be increased, there's a good chance that you have a viral infection instead of a bacterial infection. Or for example, if your basophils are increased, you're likely dealing with an allergy. So this is why we care about numbers. They actually can be diagnostic when we look at them, and it's also why it's important to know the different shapes of those neutrophils and eosinophils and basophils, because these numbers come from looking under the microscope and actually counting up the different cell types that you see.